So now we're going to look at dihybrid crosses. So in monohybrids, the root mono equals 1. For dihybrids, the root di meaning 2. So before we were looking at just one trait or one gene, such as flower color. Now we get the fun where we can cross not only flower color, but we can look at plant height. All right, so we have 2 genes that we are crossing in a dihybrid cross. All right, so let's take a look at our yellow packets, and we're going to go through the sample problem that we have in the shaded box. So our 2 traits, we're going to look at pea plants once again, and our 2 traits that we're going to look at are for seeds. Our seeds in our pea plants can either be round or wrinkled, okay? And then those seeds in pea plants can be either 2 colors. They can either be yellow or they can be green. All right, so you want to fill out your key there. The dominant is round, recessive wrinkled. Dominant for color is yellow over green. So now what we want to figure out are all of the different gamete possibilities. All right, remember gametes are sperm and eggs. All right, so to determine the genotypes, this is one of the hardest things to do because if you get this wrong, then we set up our entire problem incorrectly. So it's good to have some practice. So if you look at your yellow packet, right below the sample problem, you're going to see these 1, 2, 3, 4, 5 problems. All right, you may want to pause this and take a stab at trying to figure out what are the genotypes for these parents on your own, and then you can play it again. All right, so the first thing we want to look at is, remember, we have 2 traits here, okay? So for each gene, we have 2 alleles, so we are going to have 4 alleles total. So let's take a look at homozygous round. So homozygous, remember, same. So we're going to have 2 big R's, okay? And this parent is then heterozygous for yellow. Hetero, meaning different. So we have 1 big Y, 1 little y. All right? For number 2, we have heterozygous round. All right, heterozygous, once again, 2 different alleles, so big R, little r. And the fact that yellow can be heterozygous means that the heterozygous also applies for that second trait. So we want 1 of each of those alleles as well. All right, for number 3, heterozygous round, heterozygous, 1 of each allele. And now the only possibility for green is to have 2 recessive alleles, okay? Therefore, we know it's not heterozygous, okay? The only way we can get that trait is to have 2 recessive alleles. Number 4, wrinkled, if I remember correctly, is also the recessive trait. So the only way we can get wrinkled is if we have 2 recessive alleles. And once again, same with green, we need 2 recessive or lowercase y's. All right, and then finally, for number 5, wrinkled, once again, 2 recessive alleles, yellow, and we need to know that it's heterozygous, 1 of each allele. Okay, so that's probably one of the hardest things about a dihybrid cross is just making sure you have the correct parental genotypes. 2 genes, 4 alleles. All right, so now that we know the parental genotypes, all right, our 2 parents in this example are going to be heterozygous for both traits. All right, so here's parent number 1, all right, and then here is parent number 2. We want to determine what are the gamete possibilities. And gametes are made in a process called meiosis. And most likely we have not talked about this process yet, and we will later on. But just know that we have to think of all of the different gamete possibilities for these 2 genes. So the easiest way to think about determining the gametes, there's actually 2 different strategies. And the first I'm going to show you is called FOIL. And you probably have learned about this in math. So the first thing we want to do is remember, here's gene number 1. Here's gene number 2. And it's important in our gametes that we have one allele from each of those genes. You always want one R, one Y. And so we use our foil, and the F in foil stands for first. So we take let me make this a little bit easier here. We're going to take the first allele in each of these genes. So we're going to do a big R and a big Y. And that's our first gamete possibility. The O in foil stands for outer. So we're going to take this outside right here with this outside individual. So we get a big R and a little Y. Our I in foil stands for inner. So we're looking at this little R and our big Y. And then maybe you've guessed that the L stands for last. So that's going to be our little R and then our little Y. And for this one, it's the same, we have the same parental genotype over here, but you can practice using your foil. All right, so first is big R, big Y. Outer is big R, little y. Okay. Inner, little r, big Y. And then finally, little r, little y. Now I want to show you one other thing. There's another strategy that you can use, and just give me a minute here so I can try and erase all of this. And that is to create a Punnett square for each of the parents to figure out the gametes. So if you want to use this strategy, you get the same results. So here we go. I'm going to create a little Punnett square, okay, for parent number 1. I'm going to put my R's that I find right here on the top. And I'm going to put my second gene on the side, and then do just as we learned in our monohybrids. And now keep my letters together, which I didn't do up here. But now here are all the gamete possibilities. And as you remember, those are the same results that I got before. All right, and then you would do that for parent number 2. And in our situation, we know that it's going to be the same because they are the same genotype. So maybe this time I'll make sure that I keep my letters together. There we go. So whatever way or what strategy you like, just do that. And once again, just remembering that now these circles are going to be our gametes. So parent number 1, these are going to be our eggs from our mom, and these are going to be the sperm from our dad. Now let's take a look at what we're going to do with these gametes and how it's going to help us solve our dihybrid cross. So now we're going to take our gametes from parent number 1, and if you remember, it's going to be big R, big Y, big R, little y, little r, big Y, and a little r, little y. All right, and remember that we take parent number 2 and put those along the side. So now we have a 16 square, Punnett square. And now the fun begins. So a recommendation that I have to keep your boxes organized is to always keep the same letter first, whatever that is. All right, in my situation, I'm going to keep the R's first, and then I'm always going to put the capital letters first. So we know if there's a dominant allele that that's going to be expressed, and it's really easy to see. So this is where you kind of just sit back and enjoy this mind-numbing fun of drawing in your Punnett squares. And make sure that you're filling yours in while we're going along here. All right. And if you keep it organized, it's really easy to be able to read and interpret our results here. Okay, making sure that I'm not making any mistakes here, and I could be. All right. So now what we have are 4 different I'm going to move this up just a little bit here. We have 4 different phenotypes, okay, that are possible. So our first possibility is round and yellow. All right, and let's take a look and count how many round and yellow we have. So the first thing is we know that this is round because it has that dominant allele, and it's also yellow. So I'm going to circle that. I like using circles and different boxes to help me keep organized here. This one is also round and yellow. Okay, round and yellow. Make sure that you see why round has a dominant allele, has a dominant yellow allele. Therefore, it's going to express those. Round yellow, round yellow, round yellow. And this one's going to be round and yellow. And this one's going to be round and yellow. So if we count those all up, we should have 9, okay, that have that phenotype. Our next phenotype is going to be round and green. All right, and let's go through, and I'm going to actually use a different color for this one. I'm going to use red. So this one's round, but it's green. So I'm going to use a circle with red, and maybe I'll just go like that. Round and green. Go like that. And then round and green. It looks like that's all we have. So I have 3. And then I'm going to move on to my next one, and that is going to be wrinkled. So the recessive and yellow. I'm going to just do yellow, okay? And wrinkled yellow, I see one right here. I'm going to put a little box around it. Wrinkled yellow, and then wrinkled yellow. And I also have 3 of these. And then finally, my last category is going to be the recessive, wrinkled green. And I have 1 lonely wrinkled green. All right, and I want you to notice a pattern here. All right, we have a ratio. Our ratio is 9, 3 to 3 to 1. And I'm going to show you that. You can actually see those phenotypes on the next slide. All right, so here's what our phenotypes actually look like. So it's kind of a little bit more visual here. And once again, we have that 9 to 3 to 3 to 1 ratio. And anytime you have 2 parents that are heterozygous for both traits, just so you know, you can see this pattern, you are going to get this 9 to 3 to 3 to 1 ratio. And so you're going to start to see patterns as you start to do more and more crosses. But this is one that you want to maybe come back to. All right, so let's do practice once again. So here we want to determine the gametes, okay, the sperm of the eggs, for round green peas, all right? And our genotype for that is big R, little y, little y. So once again, if you want to do the Punnett square method for figuring out gametes, the way we would do that is we put a big R there, and put a little r here, little y, and it's kind of important to make sure you keep track of if it's uppercase or lowercase. And then we're going to easily figure out our gametes. There we go. So here are our gamete possibilities. 1, 2, 3, 4. Oops, I mixed up my R's and my Y's over there. For this parent. All right, so let's do some sample problems here. All right, so Squidward is a blue squid, and he has a genotype of heterozygous, 1 big B, 1 little b, and he's also heterozygous for a large nose. So our key here is actually big B is going to be blue, all right? And little b is going to be green. And then for large nose, we're going to use N's, and big N equals large nose, and little n is small nose. Okay, so what we also know with this problem is that Squidward's wife is a green squid, and is also heterozygous for a large nose. So we can try and figure out what is her genotype. So try and figure that out. All right, so let's see here. She is a green squid. So we know that her genotypes are little b, and she is heterozygous for a large nose. Therefore, she is big N, little n. So that is her genotype. All right, we want to figure out the gamete possibilities. So here is, once again, Squidward's genotype. We can set up a Punnett square here, or you can FOIL. You can do either or. Just going to show the Punnett square method here. So with the Punnett square method, we are going to find out that one sperm possibility is that. Ooh, and I keep doing something that I don't want to do, so I'm going to correct myself right now. Bad habit. All right, so I'm going to keep my B's. There we go. And big B, little n, and there we go. And then for his wife's possibilities, I'm going to keep my B's first. And there's only 2 different possibilities for her eggs. All right, so once again, this is what's going to go across the top of our Punnett square. This is what is going to go across our side of our 16 square dihybrid cross. So here it is set up. And once again, now we want to figure out which line or which row is Squidward's sperm. Well, here's his sperm, I believe. All right, and these are her eggs. All right, and now we want to know what fraction of their children will be green with small noses. In order to figure out and answer that question, we need to fill in our Punnett square. Keeping my letters together, keeping capitals first. And hopefully you're doing the same on your practice sheet right now. It's good practice. Big B, big B. Almost there. All right, so now green, okay, the genotype for green is the recessive, okay, so we're looking for 2 little b's, and remember small nose is also the 2 little b's, so, all right, so as you see here, we have 1 out of 16 is our fraction of children that will be both green with small noses.